Welcome back to the Great Wellness Reset podcast, where we step back from the noise, take a deep breath, and actually unpack the science behind today's biggest health conversations.
And I am Jessica. Welcome to the podcast, you guys.
Today's episode is one of those topics that people don't always ask out loud, but they're definitely thinking about. What happens to spike protein after it's produced in the body? Whether from COVID infection or from mRNA vaccination, and could it linger? If so, what are the long-term effects and how do we deal with it?
And this question has come up both in the context of COVID infection and vaccination. It's been discussed quietly in clinics, online forums, research circles, and recently in a technical article by Dr. Peter McCullough titled Dissolution of Spike Protein by Nattokinase.
Now, I'll be honest. When I first read his article, I had to slow way down. It's technical. It references enzymatic degradation, molecular structures, and biochemical pathways.
It's written for a clinical audience, but most people aren't trained to read scientific papers.
So today, we're going to review it in plain language. We're going to tell the story of how this conversation even started.
Let's rewind to early 2020. We had a brand new virus spreading globally. Hospitals were overwhelmed. Scientists were racing.
And for the first time at scale, mRNA vaccine technology was deployed. Now, mRNA had been studied for years, but never rolled out globally to billions of people in this way.
So let's explain mRNA in simple terms. Messenger RNA is basically an instruction sheet. It tells your cells how to make one specific protein. In this case, the spike protein.
Your cells temporarily produce the spike protein. Your immune system sees it, learns it, builds antibodies, builds memory cells. That's the intended design.
Here's something many people don't realize. In your body, messenger RNA, or mRNA, is normally very fragile. It's designed to be temporary. Your cells use it as an instruction sheet to make a protein, and then it gets broken down pretty quickly. The body has enzymes called ribonucleases, and that's just a fancy name for enzymes that destroy RNA. Their job is cleanup. Once the message has been delivered, they erase it.
Exactly. In normal biology, mRNA doesn't hang around. It's made, used, and cleared, sometimes within minutes to hours. But the mRNA used in the COVID vaccines was slightly modified to increase its stability. And when people hear modified, they sometimes get nervous. But let's explain what that actually means.
The vaccine uses what many call synthetic mRNA, meaning it's lab-designed rather than taken directly from a virus. It contains small molecular tweaks, specifically modified nucleosides, that make it less likely to be destroyed immediately by ribonucleases. In simple terms, it was engineered to stick around just long enough to do its job.
And that job was to produce enough spike protein so the immune system could recognize it, build antibodies, and create immune memory. If it were destroyed too quickly, you wouldn't get a strong immune response. So the added stability was intentional and part of the design, not something accidental.
Now, let's talk about the spike protein itself, because that was engineered too. It was stabilized in what's called the pre-fusion conformation. That's a big phrase. Let's break it down.
Yes, because pre-fusion conformation sounds like something from a graduate-level biochemistry course.
What it really means is that the spike protein was locked into a specific shape, the shape it naturally takes before it fuses with a human cell. By locking it into that shape, scientists made it easier for the immune system to recognize and generate neutralizing antibodies.
Think of it like freezing a key in the exact shape that fits a lock. The immune system can then study that key and remember it.
So both the mRNA and the spike protein were engineered for efficiency and immune recognition. The mRNA was stabilized long enough to produce the protein. The protein was stabilized in a form that makes immune targeting stronger.
That's important context because when people hear synthetic mRNA, they sometimes imagine something artificial or foreign in a dramatic way. But synthetic here simply means it was designed and produced in a lab to mimic a specific viral genetic instruction, with modifications to improve stability and performance.
And understanding those technical details helps ground the conversation. It explains why the design worked from an immunological standpoint, and also why questions about duration and persistence eventually entered the discussion.
Now, this is where it gets a little more complex. The vaccines were authorized under an emergency use authorization, or EUA. That means short-term safety and effectiveness were demonstrated in large clinical trials involving tens of thousands of participants. But when it comes to long-term, multi-year biological behavior, that data simply couldn't exist yet.
You can't have 5-year data on a product that's only been around for 1 year. That's just the reality of time.
Exactly. Under an EUA, decisions are made based on the best available short-term evidence during a public health emergency. But longer-term patterns, especially rare or delayed effects, can only be understood as more time passes.
And that's why long-term monitoring, or pharmacovigilance, continues. Scientists are still studying outcomes, collecting data, and refining what we understand as more real-world information becomes available.
So it's not that long-term effects were ignored, it's that they couldn't be fully known at the time. They can only be observed as the years unfold. Now, here's where the story shifts, from population data to personal experience. For the vast majority of people, vaccination followed a predictable pattern, maybe a sore arm, maybe fatigue or a low-grade fever for a day or two. The immune system activated, built protection, and life moved on. But for some individuals, the experience didn't feel that simple. A smaller group reported persistent symptoms, things like ongoing fatigue, brain fog, heart palpitations, exercise intolerance, or unusual neurological sensations such as tingling or dizziness. And in rare cases, there were more serious or even life-threatening adverse events that required medical care.
And when you're the person living through that, statistics don't feel comforting. Being told something is rare doesn't change the fact that it's happening to you. When your body doesn't feel like it used to, when your energy is different, your heart feels different, your mind feels different, the question becomes deeply personal. You're not thinking about population-level data. You're thinking, why do I feel this way? What changed? What's happening inside my body?
Exactly. And that's where this conversation moves beyond headlines and into real human experience. Because for the person dealing with persistent symptoms, the priority isn't debate, it's understanding.
It's clarity. It's answers. And it's the hope that there might be a logical explanation, and potentially a supportive path forward.
One hypothesis, though not yet proven, is that spike protein fragments, whether produced temporarily through mRNA vaccination or remaining after COVID infection, may persist in some individuals and potentially contribute to ongoing immune activation.
Researchers are still trying to figure out what's actually happening under the surface. Could it be immune dysregulation, meaning the immune system isn't calming down properly? Could it involve inflammation in tiny blood vessels, or shifts in the autonomic nervous system, which controls automatic functions like heart rate and circulation? The honest answer is, we don't fully know yet. This area of science is still developing.
So let's talk about why nattokinase gets so much attention in the conversation about spike proteins from mRNA. It's not just hype. It actually stands out for a few specific reasons.
Yeah, and the first reason is pretty straightforward. It's a protein-cutting enzyme. That means its job is to break down proteins. Since spike protein is, well, a protein, that immediately makes it interesting from a mechanistic standpoint.
Exactly. Now, that doesn't mean it automatically works the way people hope, but from a biological standpoint, it makes sense to at least explore it. It's designed to cut proteins apart.
And what really put it on the map in this discussion was the laboratory research showing that it could degrade spike protein in vitro, meaning in a controlled lab setting.
Right. In vitro just means in a test tube or cell culture, not in a human body. But it showed that spike protein isn't indestructible. Under the right conditions, nattokinase was able to break it down.
And that's important because it demonstrates vulnerability at the molecular level. It tells us the spike protein can be enzymatically degraded.
But nattokinase isn't only interesting because of that one lab study. It also has documented systemic fibrinolytic activity in humans.
Let's explain that word. Fibrinolytic.
Yes, please.
Fibrinolytic means it breaks down fibrin. Fibrin is the structural protein that helps form blood clots. So, nattokinase has been studied for its ability to support healthy circulation by breaking down excess fibrin. And COVID infection was associated with clotting abnormalities in some patients. So, nattokinase was already being discussed in circulatory contexts.
And that's a big deal because most enzymes people are familiar with, like digestive enzymes, mainly work in the gut. Nattokinase is different. When taken properly, it can have systemic effects, meaning effects throughout the body.
So, it's not just another digestive enzyme helping you break down food. It's classified as a systemic proteolytic enzyme, which simply means it can circulate and cut down proteins. Proteolytic enzymes act like molecular scissors. And on top of all that, nattokinase isn't some brand new lab-created compound. It's actually derived from natto, which is fermented soybeans, a traditional Japanese food that's been consumed for centuries.
Right, and from that fermentation process comes nattokinase, which is what we call a proteolytic enzyme, meaning it breaks down proteins.
And because it's been used as a supplement for years, especially for cardiovascular support, we actually have a decent amount of real-world use data behind it.
So when you put that together, traditional origin, protein-cutting activity, lab evidence of spike protein breakdown, human data showing fibrin effects, a track record of supplement use, and a long history of use for circulation, you can understand why it gets attention in conversations like this. Not because it's proven to solve everything, but because biologically, it makes sense enough that many clinicians think it deserves thoughtful consideration. In 2022, Tanikawa and colleagues published a study in the journal Molecules. They exposed spike protein to nattokinase in vitro, meaning in a lab dish.
And they observed that the spike protein degraded over time and with higher enzyme concentrations, meaning the enzyme broke it down.
That's significant from a structural standpoint. But, and this is critical, this does not prove that oral nattokinase dissolves spike protein in humans.
Even though it's lab data, it shows spike protein isn't indestructible. It's susceptible to enzymatic breakdown. That's biologically interesting.
It opens the door to the question. If nattokinase works systemically in humans, could it theoretically contribute to protein breakdown beyond fibrin?
Now let's spend a little more time on the human data, because this is important. In 2015, a study by Kurosawa and colleagues looked at what happens when people actually take nattokinase orally. They gave healthy participants a single dose of 2,000 fibrinolytic units. That's how nattokinase activity is measured.
Right, and fibrinolytic units basically measure how effectively the enzyme can break down fibrin, which is the structural protein that forms blood clots.
Exactly. So what they did was measure certain blood markers before and after the dose. These markers included things like D-dimer and fibrin degradation products. Now, let's simplify that. D-dimer is basically a fragment that appears in the blood when fibrin is broken down. So if D-dimer levels rise, it suggests fibrin breakdown is happening.
And what they found was that within hours, we're talking about 4 to 8 hours, those markers increased significantly. That tells us something very important.
Yes, it tells us that nattokinase, when taken orally, isn't just sitting in the digestive tract breaking down food. It's having measurable effects in the bloodstream.
That's a big distinction. Most enzymes people are familiar with, like digestive enzymes, mainly work locally in the gut. But this study suggests nattokinase can exert systemic activity, meaning effects throughout the body.
Now, we should clarify something here. The Kurosawa study wasn't studying spike protein. It was studying fibrin and clot-related markers. But what it does show is that nattokinase survives digestion to some degree and influences measurable biological processes after ingestion.
So from a scientific reasoning standpoint, that becomes part of the larger discussion. If an enzyme can reach circulation and break down a structural protein like fibrin, then it raises the question, could it also interact with other proteins under certain conditions?
Again, that's not proof of anything specific,
but it establishes biological capability. And in science, understanding capability is often the first step before clinical outcome studies are done.
Exactly. It's about understanding what's possible based on mechanism, and then asking the next question responsibly. All right, that's a lot to take in.
It really is. We just covered vaccine design, enzyme mechanisms, lab studies, and human data. And that's not light material.
So let's pause for just a moment, take a breath, and then we'll come right back to continue breaking this down in a practical way.
Stay with us. We'll be right back.
If you're looking into systemic enzyme support, Pro-Onezyme by Wellness One brings together Nattokinase along with protease, C-Pros-S, papain, bromelain, peptidase, catalase, and other complementary enzymes. This thoughtfully designed blend is created to support healthy circulation, overall protein balance, and a well-regulated inflammatory response. You can learn more about Pro-Onezyme at procell.com/wellness1 or find it on Amazon by searching Wellness One Pro-Onezyme.
All right, we're back, and let's pull everything together for a moment. In controlled laboratory settings, researchers observed that nattokinase was able to break down spike protein. That tells us the protein is susceptible to enzymatic degradation under the right conditions. In human studies, we've seen that nattokinase can enter circulation and measurably influence fibrin breakdown, meaning it has systemic biological activity after ingestion. What we don't currently have are large-scale, randomized clinical trials showing that nattokinase definitively clears spike protein in people. That distinction is important as we continue evaluating what's known, what's plausible, and what still needs further investigation.
That's the honest position.
So this naturally brings up another question. Why combine enzymes at all? Why not just use nattokinase by itself? Because in many systemic enzyme formulas, nattokinase isn't used alone. It's paired with other enzymes that each play a different role in how the body manages proteins and inflammation. You'll often see things like protease, which is a broad term for enzymes that break down proteins in general. C-Pros-S, a specific protease studied for supporting protein turnover and inflammatory balance. Papain and bromelain, plant-derived enzymes from papaya and pineapple. Peptidase, which breaks proteins down even further into smaller fragments. And catalase, an antioxidant enzyme.
And this is where it gets really interesting, because the idea isn't that one enzyme does everything. It's more like a coordinated effort. Think about it this way. If your body is dealing with excess or damaged proteins or inflammatory debris, that cleanup process doesn't happen in one step. First, larger proteins may need to be broken down into smaller pieces. That's where proteases come in. Then those smaller pieces may need to be broken down even further. That's where peptidases help.
Exactly. It's like dismantling a building. You don't just knock it down in one swing. You break it into sections, then into smaller pieces, then into manageable fragments.
And C-Pros-S is interesting because it's known for its ability to help break down non-living protein material while supporting a balanced inflammatory response. In simpler terms, it may help the body process leftover protein fragments without overstimulating the immune system.
Papain and bromelain add another layer. Most people think of those as digestive enzymes, and they are. But beyond digestion, they've been studied for their role in modulating inflammation and supporting tissue recovery.
Now let's talk about catalase, because that one's different. Catalase doesn't break down proteins, it breaks down hydrogen peroxide, which is a reactive oxygen species produced during inflammation.
In other words, when there's inflammation happening, oxidative stress often increases. Catalase helps convert hydrogen peroxide into water and oxygen. It's basically a cleanup enzyme for oxidative byproducts.
So when you look at this whole combination together, it starts to make more sense. You've got enzymes helping break down larger proteins, others helping refine those fragments, others supporting inflammatory balance, and others supporting antioxidant defense.
Instead of targeting one single molecule, the philosophy behind combining enzymes is to support the body's overall cleanup systems: circulation, protein turnover, and inflammatory regulation in a layered way.
And that's why many integrative practitioners find the combination approach compelling. It's not about one magic ingredient. It's about supporting the body's natural processes from multiple angles.
It's more like assisting the body's housekeeping staff, rather than trying to bulldoze one specific target.
Exactly. Support the systems, not just the symptom.
This is where the integrative perspective comes in. Some integrative clinicians look at this and say, if there's biological plausibility, meaning the mechanism makes scientific sense, if there's lab evidence showing the protein can be degraded under certain conditions, and if the compound in question has a generally favorable safety profile when used appropriately, then it's reasonable to have the conversation. Not as a conclusion, not as a guarantee, but as a discussion point.
And that distinction really matters. There's a difference between saying something is biologically plausible and saying it's clinically proven.
Exactly. Plausible means the mechanism lines up with what we know about biology. Proven means large human trials have confirmed the outcome. Those are two very different levels of evidence.
So when integrative practitioners talk about enzyme therapy in this context, many aren't declaring it a cure or a certainty. They're saying, given what we know about how these enzymes work, it may be worth exploring thoughtfully, especially when monitored properly.
And that's different from hype. It's about measured curiosity. It's about recognizing that science often begins with mechanism and small studies before larger trials catch up.
So the integrative stance isn't, this solves everything. It's more, there's enough here to warrant careful consideration and further investigation.
And in complex health situations, sometimes that middle ground, thoughtful discussion without exaggeration, is the most responsible place to stand. When you zoom out, science rarely moves in straight lines. It builds step by step, one study leading to another, one question opening the door to the next.
And the discussion around spike protein persistence and enzyme therapy is part of that process. It's still developing.
Over time, we'll get clearer answers.
Whether large institutions choose to invest heavily in researching this specific area remains to be seen.
For people dealing with persistent symptoms and still looking for answers, it makes sense to explore options, especially when there's some biological reasoning and early evidence behind them. If a strategy has plausible mechanisms, supportive lab data, and a history of supervised use, many feel it's reasonable to consider it rather than simply waiting and doing nothing. And that's where a nattokinase-based systemic enzyme blend enters the conversation. Instead of focusing on one isolated compound, some clinicians look at a broader enzyme approach, combining nattokinase with other proteolytic enzymes that support overall protein turnover, circulation, and inflammatory balance. The idea isn't about targeting one single molecule in isolation, but about supporting the body's natural cleanup systems in a layered way.
Right. And the key word is support. A systemic enzyme blend that includes nattokinase works within normal biological pathways that already exist in the body. It's not forcing something unnatural. It's assisting processes like protein breakdown and circulatory balance that are already happening every day.
Exactly. When there's some mechanistic support and real-world use data behind an ingredient like nattokinase, especially when it's part of a thoughtfully designed blend, it becomes something that many integrative practitioners feel is worth a measured conversation.
And that conversation should always be informed. It's about understanding your personal health picture, making sure it fits with your overall goals, and approaching it in a steady, reasonable way.
Curiosity paired with caution, and a focus on supporting the body's natural systems rather than chasing quick fixes.
If today's episode helped you take a complicated, highly technical topic and understand it in a clearer way, share it with someone who might need that clarity. These conversations are often filled with noise, and sometimes what people really need is context and balance.
And if you want to go deeper, we always encourage you to read the original source material for yourself. Dr. Peter McCullough's technical article, Dissolution of Spike Protein by Nattokinase, is available online. It's detailed, clinical, and data-heavy. And it's worth reviewing if you want to see the raw scientific discussion behind what we unpacked today.
You can dive deeper into topics like this at greatwellnessreset.com, where we break down complex science into clear, practical insights you can actually use.
Be sure to follow the Great Wellness Reset podcast so you don't miss our upcoming deep dives. We're committed to bringing thoughtful, balanced conversations to every episode.
You can find Pro-Onezyme with Nattokinases at procell.com/wellness1 or on Amazon by searching Wellness One Pro-Onezyme.
Thanks for spending this time with us. Stay curious, stay informed, and keep pursuing better health resets. We'll see you next time on the Great Wellness Reset.